talk about one extra lecture to instead of last week. And 2 weeks back, we didn't have any lecture because I have many lectures with many cities. Students sometimes I'm confused. But you have only one lecture, so easy to remember what did you read and what did you hear. 2 weeks back, we didn't have lecture, yes? Okay. Okay. So now we start. So we want to talk about microbial ecology. What about microbial ecology? So because mostly we think about biology, and biology has different parts, different branches. One part is ecology. Like most courses which have logy, like bacteriology, hematology, physiology, and many others, logy means understand, try to know, have a knowledge. And ecology, we want to know, we want to understand about living things, relationships with each other, and with environment. So suppose first some examples regarding to the animals, human, and plants. Suppose if I'm in an area near river, so river affect me. I can have agriculture, I can have some domestic animals there. If I'm living in a desert, in the Sahara, it affects me. So non-living things affect living things. And living things affect non-living things. For example, I make some barrier in front of the river. I have some factories. I make the air polluted. Some pollution in the river, some pollution in the soil. So, living things, environment, and environment affect living things. And living things affect each other. A lion eats, for example, one sheep eats plants. So they affect each other. Sometimes they help each other. Some wolves come with each other to have a predation. So wolves, group wolves, it is a population, and all of them help each other. So what is ecology? In ecology, we want to know the relationship between living organisms with each other, and the relationship between living things and non-living things, and the relationship between living things and non-living things. It is ecology. So living things are plants, animals, fungi, protists, bacteria. So in this course, we want to focus only on microbial ecology. We want to know what are the relationships between microbes with each other, between microbes and environment, and to understand what are these relationships. So in this lecture and course, we focus on microbial ecology. Microbial ecology. But we knew ecology of plants and animals because we can see them very easily by unarmed eyes. But for understanding regarding microbial ecology, it was needed to have invention of microscope. So first, right now, we want to have one introduction. In half an hour, we want to have an introduction before starting microbial ecology. Okay. The beginning of microbiology. Beginning of microbiology. So the great scientist with the name Anthony van Leeuwenhoek. Anthony van Leeuwenhoek. He was a Holland scientist in 60s. We use diversity of previously hidden mold of microbes from natural habitat. So he used the simple microscope and he could see the microbes, and he had some sample from soil, from water, from air, and he understood there are some microbes, microscopic organisms. They are living things, and he had some examination about them. He had the earliest description of bacteria and the earliest studies on microbial ecology. For example, he saw some microbes are bigger, some are smaller. Some have cilia, some have flagella, some are predator, eat and engulf the other microbes, and he wrote whatever he observed. On that time, it was a hypothesis with the name of spontaneous generation. Spontaneous generation. What is the meaning of a spontaneous generation? It means one living thing coming from nothing. What is nothing? One living thing come without parent. So right now we know it is impossible. Any living thing has parents or parent. Those which have sexual reproduction, each living thing has parents. And for bacteria, for virus, many microbes have only one parent. They come from binary fission. 1 to 2, 2 to 4, 4 to 6. So on that time, they believed if you put a water on a table, on a glass, in a glass on a table, after some time without any parent, many microbes are there in the water. If there is a soap on a plate, on a table, after some time without parent, many microbes will be there. So the first scientists want to remove this hypothesis was Lazzaro Spallanzani, Italian naturalist, debating the view of spontaneous generation. What did he do? He put some liquids in different flasks, boil it. So by boiling, he tried to kill the microbes, and he could. Then, after finished the boiling, closed completely the door of the flask. And he explained that microbes coming from air to a liquid. If you kill the microbes in the liquid and you close the door, and the new ones cannot come inside, so there is no spontaneous generation. There is no spontaneous generation. He showed that putrefaction of organic substances is caused by minute organism. If you put a meat on a table, so a small number of microbes come from air to the meat, they come from air to the milk, they come from air to the food, and they multiply. So each microbe has parent. They don't come from nothing. They don't come from without parent. So showing that putrefaction of organic substances is caused by minute organisms that don't arise spontaneously, but multiply by cell division. He showed heating destroys these organisms after killing them. Sealing or closing the containers then prevent spoilage, but it was indefinitely because of heat-resistant bacterial spores. It has one problem. Scientists believe that when you close completely the door of the flask, oxygen cannot go through the food, and a spontaneous generation coming without parent, it is dependent on the presence of oxygen. It was not wrong, but any question should be answered. Any question, wrong questions and correct questions should be answered. We cannot say it is wrong question and I don't have any answer. And another problem that he was successful because those liquids he used didn't contain spore. You know, some bacteria like Bacillus and like Clostridium can produce a resistant form with the name of spore. And whatever you boil it, you cannot kill them. So if there is a spore and you boil it, you cannot kill them. After sealing, after closing the door of the flask, again they can grow. And those scientists believe that there is spontaneous generation. The other scientist coming to solve the problem was Louis Pasteur. He was Louis pastor, 860 to 862. Demolished the theory of conservation by cultural ethics. Organisms in day and the road in replaced water, sugar water, urine, sugar-based juice, water, different flasks. Then we shed the net of the flasks, not stealing, not losing. So told other scientists see oxygen very easily can come to the liquids. There is no oxygen problem. So we boil this liquids and heal the boil and come inside the liquid. This is interesting. This is Institute in Paris. So till the end of the life, where microbes cannot reach the liquid, according to the leg, the lemon, and the new microbe can be present in the liquid. Louis Pasteur was lucky because those so after this experiment, some scientists showed that another liquid this rule not to be approved. Other scientists, after some time, found on Julius Kahn in 1886. This is right and for scientists understood what endospores are. And the last scientist who invented the spontaneous hypothesis completely, it was John Tyndall. In 1893. So, Pasteur and the other scientists couldn't kill spores by boiling because spores are heat resistant. So, John Tyndall used another method and called it Tyndallization. What was that? John Tyndall used several cycles of germination of endospores and destruction by subsequent heating. What does it mean? He boiled it, so he killed all vegetative form, not spores. Again, stop heating. So some spores believes the situation is okay. So they change their appearance to vegetative form. Again, heating, kill vegetative, spores remaining. Again, stop heating. Again, some spores, some remaining spores believes situation is okay, changing to vegetative. So by treating spores by many times heating, not heating, heating, not heating, boiling, not boiling, boiling, not boiling, all the spores of the liquid, and by the device he showed that spontaneous generation is not correct. Reliable sterilization of all media at 100 degrees centigrade. At that time, they didn't have autoclave. In autoclave, your temperature is 121 degrees centigrade because it is with high pressure, 15 pounds per square inch. Yes, it is 15 pounds per square inch. 1 second. And 121 degrees centigrade. It is autoclave. But at that time, they didn't have this device. They didn't have autoclave. So, by tyndalization, scientists from that time could kill all spore formers in any liquids. And they showed in microbial ecology, which any microbe has parent. Without parent, there is no new microbe. Okay, is it clear? Can I go to the next part? This is history. Maybe you knew it before coming to the class from previous lectures. So we have some introduction. We complete the introduction and we go to the main part of the course. Okay. So at first time when microbiology started, it was only liquid media. It was only liquid media. And there was a problem. In liquid media, any sample from soil or from water, they contain mixed cultures. Mixed cultures. So many bacteria, many protozoa were in one medium. So in order to understand them, in order to check them, in order to know many things about them, it was needed to make them separate. Separate. So we call it pure culture. So we have two cultures, mixed culture and pure. Mixed culture and pure culture. Pure culture means in one medium there is only one microbe, one specific microbe. For example, in nutrient plus only you have Escherichia coli, or in one tube you have only Klebsiella pneumonia. But mixed culture, suppose there is a sample from stool or from sputum. So when the sample you use for culturing, many bacteria can grow. You should make them separate by pure culture. Pure culture. The ability to determine the etiology, cause of disease, depends on the development of appropriate techniques and methodological approach. So one person has abdominal pain, with diarrhea, with loose motion. So when you have culture from stool, there are many microbes. You should separate them and understand. Is it Salmonella there? Is it Shigella there? So it is done by pure culture. So by pure culture in medical microbiology, you can understand the etiology. What is the etiology of disease? Or what is the cause of the disease? What is the cause of the disease? So the first person think and understood how he could pure culture was great scientist with the name of Koch. 1883 developed a simple method for isolation and maintenance of pure microbial cultures on solid media. So from that time, we have two types of media. One media are liquid and one media is solid. So which one is better? The question is not complete. When we say which one is better, we should ask in which situation. If we want a medium for isolation and purification, solid media is better. So if our purpose, if our aim is isolation and purification, so solid media is better. Solid media are better. But if you have pure culture, you want it has maximum growth, maximum enzyme production, maximum motility. It has the best situation. Best situation is in liquid media. So best growth is in liquid media. But for isolation and purification, solid media are used. At first time, solid media they were very simple. First, he used slices of boiled potato, and he had some simple colonies or this boiled potato. Afterwards, he found gelatin, and later on, agar was found for solidification. For example, when we say nutrient broth, it means it is a liquid media. Yes? Nutrient broth. It is liquid medium. But if we say nutrient agar, it is solid medium. Broth, nutrient agar. And we can buy nutrient broths and agar. When we want to have liquid medium, we use nutrient broth. When we want to have solid medium, we use nutrient broth, and we add 1.7 or 1.5 gram it doesn't write properly. One second. One point five gram of agar in 100 milliliter of nutrient broth, make it solid. 1.5 gram per 100 milliliter of liquid medium, make it solid. It allowed microbiologists to separate a microbial type from heterogeneous community. For what? When you have one microbe in one plate or when you have one microbe in one tube, so you can easily study its activity. If 100% doing music, so in between 100%, how can you understand which one is playing well? So separate, you should hear. If all the students with each other talk to the professor, the person cannot understand anything. One by one should explain what they want to say. So each microbe should be purified. And after purification, it is easy to study their activity. And their complex interactions removed from observation. Many of the pure culture techniques devised by Koch are being used today with modification. Observation of first interaction between Penicillium contaminant on Staphylococcus aureus culture observed by Sir Alexander Fleming, 1929, actually yielding life-saving antibiotics. So one of the most observations in microbial ecology was reported by Sir Alexander Fleming. He was working on a bacterium with the name of Staphylococcus aureus. He had a trip going and come back. After the trip, saw some colonies of fungi were in the plates, and there were inhibition zones between fungi and bacteria. This inhibition zone was a zone which fungi or mold by production of chemical substances could kill those bacteria. Okay? He could understand that these chemicals can be useful. And this is the observation of microbial ecology. What is microbial ecology? The interaction between microbes. Yes, it is the interaction between two microbes. One microbe produces a chemical substance and could kill another microbe. It is an interaction. Yes, many scientists believed that it was by accident and it was by chance. That he answered, during your life, you have more than what I have chance. That God gives some people good mind and good eyes to see the chance. Many times your chance is in front of you, but you cannot see them. So it was the answer of Alexander Fleming. Okay. So . technology in the 20th century and in our century right now began to establish the role of microbial metabolic activities in global ecological processes, the role of microorganisms in the biodegradation of organic substances. Suppose when spring and summer finish and we go to the fall or autumn, many leaves, many woods from trees come to the soil. 1 second, suppose during many million years, if there were no microbe to degrade these leaves, what happened? So all the earth was completed with many garbage, and microbes change the organic matters to the minerals and recycling. So plants again can use it. Plants again can use these substances. Suppose when an animal die, when a human die, when a tree will die, microbes degrade them and recycle them to the soil, to the mineral matters, and again can be used by the trees, by the plants. 1 second, suppose all human and animal and plants which died during million years, it was no microbe for degradation. Now all over the world was full of many dead people and dead animals and dead plants, and it was no minerals, because minerals changed to the organic matters and organic matters didn't recycle to the soil. So they are very helpful. Microbes in the soil are helpful, and sometimes we can use them. Suppose we have factory, we should have factory, it is important to have factory, but sometimes our factories have pollution to the air, have pollution to the soil, have pollution to the water. They have many pollution. Suppose Iran and Iraq have oil, and sometimes they transfer by some ships to the another countries. So Persian Gulf, Oman Sea, the Indian Ocean are polluted with oil. So what should we do? Who is responsible for removing the pollution? So those countries which produce pollution are responsible for removing it. Some people believe because 2/3 of the earth is full of water, so it is no need to clean the ocean. There is no need to clean the Persian Gulf. There is no need to clean the Oman Sea. But it is not true, because from this water we receive our fish to eat. In this water we go to sightseeing, to sit beside the ocean. So it should be clean, and it is for our children in the future after 100. So we should clean it. But human cannot clean it directly. We can find good microbes in the water with capability of degradation of oil, take it to the lab and multiply, help them to multiply, elevate their numbers, and then add it to the places which are oil are present in the water. So they work for us without any salary. They don't ask you any money. They don't ask you any sleeping time. Only they work and work for cleaning. So microbes, a few of microbes are pathogens. Most microbes are good, like people. Most people in a country are good people. A few of them are bad. So it is normal. But if a few persons are bad, we cannot say all the people in the country are bad. No, it is not correct. So most microbes are good. Microbes help people more than people help microbes. What did we do for microbes? But microbes all the day help us in agriculture. Microbes produce insulin for us. Microbes are producing anti-cancer for us. Microbes degrade pollution. Microbes in agriculture produce nitrate. Microbes can be used in biocontrol. So who is friend? Microbes are our friend, but we don't do anything for our friends, only we use them. Okay? So microbial ecology is not only a pure science. It is the first step for practical works. If some microbes are enemy, we should know them, to stand in front of them. If some microbes are good, we should know them and understand how can we use them. So microbial ecology help us to know our friends and to know our enemies in the environment and try to use them. Suppress the pathogens. If you have agriculture in between two rivers and there are some pathogens which infect your plants, how can you suppress them? For suppressing them, first you should know them. So it is microbial ecology. First you should know what is the interaction of the microbe with another microbe, pathogenic microbe and plant, and how can you suppress it? If there are good microbes in the river, if there are good microbes in the soil, you should know them. How can you know them? By microbial ecology. So microbial ecology is first step before biotechnology. Before biotechnology. So when you understand which microbes can produce antibiotic, you make a factory and you produce antibiotic from it. So it is microbiology. When you know which microbe is pathogen and how is its pathogenicity and how can invade you, you can make vaccine. So production vaccine from a pathogen, it is biotechnology. So microbial ecology is the first step for going through the biotechnology in agriculture, biotechnology in medicine, biotechnology in medical processes, biotechnology in bioremediation, in cleaning the ocean, cleaning the soils, can be helpful. And sometimes some scientists show us, for example, preparing a column containing different substrate and showing in each part different microbes are living and have many interactions with each other. It is by your like. If you want, you can check in the internet Winogradeski column. Beijing, another scientist, studied on microbial Daydream found a non-symbiotic aerobic nitrogen fixing and isolation of sulfate reducers. So Daydream understood. Daydream looked at the farmers. He saw that farmers, after harvest the beans, they take the roots of plants, cut it, and put it in big plates containing water, and put beans in cutted roots, and after that put in the soil and sowing, and it grows very well. Those people working in agriculture knew it is good, but they didn't know. They didn't know what are they doing. They did microbiology. So symbiotic microbes were present in the roots, in the nodules, and they cut the nodule and roots. So bacteria come to the water, and by the beans inoculation, microbes can penetrate to the bean, and after sowing, bean was grown very well. It is like a fertilizer. You see, agriculture people buy nitrate, phosphate fertilizers. So these microbes work as a fertilizers. Many years, many hundred years, agriculture did it. Yes, of course. Bacteria that live in the roots of legumes. So people, before understanding microbiology, use the microbes in biotechnology. So people, before knowing what is the biotechnology and what is the microbiology, they did microbiology and biotechnology. So they drink finding some microbes are good for plants, and some of them are symbiotic. They live in the roots, and some of them were around the roots, inside the roots, around the roots, and they work as a fertilizer. Based on his principle, everything is everywhere. The environment selects. What does it mean? Everything means microbes. He wants to say microbe is everywhere. The environment selects. Suppose, as much as you look on the table, you cannot see any fungus. You cannot see any mold. Excuse me 1 second. Excuse me. So he says microbe is everywhere. What does it mean? Everything you look on the table. There is no thing. That fungus are inside our home, are in the environment. If the environment allows us to behave themselves, see, you put a refrigerator. Don't put it. Put it on the table for one day. So when it is in the refrigerator, by cold situation, you don't know that the microbes grow. But when you put it on the table, now the environment allows them to grow and you can see them. See why? Wash your hands. Why do you wash many times during a week your body in the bathroom? Because you know microbes are around you. If you make yourself clean, and if you live in a hygiene place, so this good environment not allow them to show themselves. That if one person don't wash body, don't wash hands, don't brush teeth, you can see the risks are with decay and somehow coming to the skin, some bacteria can coming, so microbe is everywhere. The environment select. You change the environment in a manner they cannot show themselves. They cannot. Why do you ask in this case? So, why don't you go to the crowded places? If you go to the crowded places, where you wear a mask, so coronavirus is everywhere, especially in crowded places, and if you don't have a mask, and you be there for a long time, everyone should show themselves. And when they show themselves are sick, you have diseases, you have problems. Then we are brothers to go heritage of heritage culture. Some microbes are easy to grow. But some microbes cannot produce, they have problems in their metabolism. They can produce some organic matters. For example, one microbe cannot produce some amino acids. So you should receive it from the environment. If you want to develop them, you should add those problems they have to the material. If the microbe cannot produce two amino acids, you should put it in the environment to add it to grow. Why do we have why? Because some are easily growing, some are facilities. Facilities hormones need more food than our microbes need. For example, you have two children. Whatever you put on the table, one of them eats and says thank you. And another child says, I don't like fish, I don't like meat, I don't like pizza, I like for example, chocolate. So he's fastidious. And you try to manage his diet in a better way. But microbes, you cannot manage them. It's according to genetics. Some easily grow on any media. Some are facilities. we use blood agar, serum agar, chocolate agar, and we call them enrichment culture. They are enrichment culture. Sources of information by microbial ecologists. What are the sources of information? So wherever there is ecosystem, wherever microbes are present, it is the source for microbial ecologists. Like what? So one is microbiology of soil. So soil is full of different microbes with different interactions between them and different interactions between microbes and environment. So one source is microbiology of soil. Another one is medical microbiology. Medical microbiology. So here we want to understand what are the interactions between the pathogenic microbe and human. What do microbe to human? What is the differences of human to microbe? And how can we help human to win the game, the war? It is a war between pathogen and human. When we want to know microbial ecology about it, because we want to help our friends, our family, our other humans to stand. So microbial ecology in medical microbiology, it is not pure science. It is the first step for practical science. Microbial plant disease. So in most countries, the first thing is important is agriculture. Because before thinking, before talking, before going, before sleeping, we need to have food. Whatever we do, we need energy. Without energy, we cannot do anything. So the first aim of one person of going to work, going to university, and going to everywhere is the food. Every day, 3 times we have food. In between, again, we have food because ATP should come to my brain to think. ATP should come to my heart to blood pumping. ATP should come to my lung to breathing. After thinking and blood pumping and air coming to my lung, I can do another things. So food is very important, and especially understanding the interactions between microbes and plants. Which microbes are beneficial and we can help them to grow plants better. Which microbes are pathogens? Which ways do we have to stop them, to suppress them, and not allowing to reduce plant production? Freshwater microbiology is very important. So 2/3 of the water of the world is salt water in oceans and seas. A small quantity of the water in the world is fresh water, and we can drink it. So if we have a river, if we have some wells, and we have underground water, before drinking, we should know the ecology of the river. What is the ecology of the underground water? What is the ecology, microbial ecology of the lakes? So after knowing it, we can understand what can we do for purification, disinfection, and sell it for drinking. Another thing is sewage disposal. So many sewage coming from human, from cities, from villages. So it contains many organic matters and water. So it should come to the environment. But how? Can it come directly to the river? So first we should know the microbial ecology of this disposal, this sewage. First we should know. And for example, there is a sewage full of bacteria. First, which bacteria are there? Second, which bacteriophage or viruses can kill those bacteria? And how can we use them? And we should reduce the bacteria and then can send it to the environment. I told you, if we produce the pollution, we have the responsibility to remove the pollution. So we should understand the microbial ecology, and before sending sewage disposal to the environment, to the river, to the sea, to the ocean, to the soil, we should minimize the microbes and pathogens there. Marine microbiology. So 2/3 of the world is marine and ocean, and there are full of different microbes. When we know them, we can understand which help they can give us. Can they produce antibiotic? Can they produce anti-cancer drugs? Can they use as a food for animals like algae? Can they use as single cell proteins for food additives and the others? So before using the large number of microbes in the marine, first we should know them and understanding what are their interactions. Food microbiology. Food microbiology. So food is important and nobody has another idea. Food is very important in our life. So when we go to agriculture, from the time of sowing, from the time of growing, from the time of harvesting, from the time of packaging, from the time of putting them in supermarket, in shops, from the time to have it at home before use it. So all these times, many microbes can attack the food. We should know during the agriculture, during harvesting, after harvesting, during delivery, which microbes have problem to food. So it is microbial ecology. And how can we stop them? How can we suppress them? So from pure science to practical science, from knowledge to action. When we know them, then we can have program. Then we have saying what should we do. For example, some microbes have problem to our food. Some foods are drying, like some fruits. Sometimes microbes have problem. We have some our food salting. For example, salt in very cold places, they have salting fish. So microbes cannot go with high salt. So before eating, many times washing and washing till salts going out from fish, and then cooking and eating. When we understand, for example, Canned food may contain this clostridium botulinum. We boil, we heated the canned food. So if we do something, we do after knowledge, and knowledge is microbial ecology. When we know them, we have a program how to suppress them, how to stop them. Okay? Okay, a few minutes, maximum 5 minutes, we talk on this page, and then we finish. So, introduction finished. Right now, we talk regarding to this photo. What is this? Sometimes we talk about one member, one person, one animal, one microbe. So it is individual. When it is alone, individual. If the same microbe are in the same place, the same microbe in the same place, we call it population. We call it population. If there is two different population, it was better, for example, blue dots and green dots. Yes, make this green. It is community. Two different, for example, paramecium and didinium. So paramecium is eaten by the didinium. Didinium is predator. Didinium is the predator of paramecium. It is a community. If at least we have two communities together, we have the simplest ecosystem. So the same members in one place, population. Two populations in one place, community. At least two community in one place, it is ecosystem. So what should we want to do? So we want to talk interaction. So one doesn't have interaction to itself. So when we have population, we have interaction of number 1 to number 2, and we have interaction number 2 to number 1. So next time we talk about interaction between or among members of one population. And after that we go, what are the different interactions between one population to other different population? One population to other population. What are the interactions present between these two groups? So first we talk about interactions inside one population. Then what are the interactions between two different populations? Between two different populations. Okay, so would you please, I request you, next time in next lecture, you remind me. We are in page 5 because I have different lectures with different students from different cities, and sometimes I forget because you have only one ecology, but I have several ecology. So maybe sometimes I'm confused. Which page should I start? So would you please next time tell me we finished page 5 and we should start page 6. This is number 1. And the other thing that, as you know, next week is the Iranian New Year, and in Iranian New Year lectures are off till end of the holidays. So we will continue after the holidays. The second thing, please remind me, after the holidays, remind me which I missed one lecture according to the conference. So in 1 week, we have twice lectures, and I have 2 groups of ecology. So one group, one lecture is front, and another group, you are one lecture, you are back. So after the holidays, both groups should come together to one position regarding to the lecture. So please remind me after the holidays. And I hope you have a good times. Holidays and Eid is regarding to those which we are kind to the people and people are kind to us. So it is Eid. And if you are not kind, so it is not Eid. And as we see microbes in the next lecture, we can see microbes help too much each other, and we believe we are greater, we are human, we are the most best creature which God created. So we should show we are the best, and we do it. Okay. Have a good time with your family. I hope health and happiness. See you after holidays, inshallah.